to week 8. Let's get started. Our topic this week, of course, is biological sciences, and we are pretty much looking at breaking this topic into two parts. It's quite a large one, and we're going to look at how our biology can actually be approached in the classroom. So to get started, well, let's get started. The first thing we can talk about is the fact that there's many possible ways to study biology. Now, the text we've referred to is Loxley this week, and Loxley does a fabulous coverage of classroom biology, and I do recommend that you get in there and read the relevant chapters. But we can use textbooks, we can use websites, television shows. I mean, we've got, all sorts of different things, podcasts on the ABC, newspaper articles, all assume that, a certain level of knowledge in biology. So they're discussing environmental issues, loss of habitat, endangered species, agricultural and food issues. And we know, for instance, there was a report out yesterday through the ABC and CSIRO who are talking about species degradation and loss, and talking about figures in excess of 40% up in the northern tropics. So we're talking about a whole range of ways of accessing science. What I'm going to argue this week, though, is science can be a black hole. I mean, we can actually lose ourselves by exploring things in science that are not relevant to us, and by chasing interest or engagement. Whereas actually, if we bring it back to our core, back to where the students are located, we may be able to set up much better inquiry processes. So once the spark of excitement wears off when you're teaching science, you really need to be working with the long, slow burn of engagement, and that's what we're trying to work at here. So what I'm recommending this week is we keep it local. And if we are doing and working with science, that we build from that point. And there's a couple of projects that have been completed, one out of Deakin University that identifies 3 hotspots in any school area. Invertebrates in the school ground. You can build a really detailed study, a unit of study around that. Studying the adaptive behaviour of small animals in the classroom. And finally, looking at plants in the school environment. And this is the way we're going to structure this week so that we can actually explore it. Carey and Steppens in 1985 found that movement or action is the main criteria that children use to determine if an object is alive. Now, when we start teaching biology, I mean, clearly if we go back to the ACS, we see that it's got to do with living organisms. And when we talk about living organisms, I mean, we assume we know what's living, we assume we know what's dead, but in fact, or non-living. But the question here is we're really working with our students, with the learners' concepts. And what we need to do is actually work out what they understand as living and non-living. And this, you're going to find, is going to vary incredibly as you move through the curriculum. For instance, how often do you hear in a play school or early childhood, or even grades 1, 2 and 3, the sun is hiding behind the cloud? I mean, as teachers and as educators, and even as parents, we talk in metaphors. And often language can actually work against developing and supporting student conceptions of science and scientific frameworks. We look at butterfly life cycles as a really interesting one. Young children, for instance, did not identify eggs and pupae as alive or life, and yet we're able to identify butterflies as life. We look at eggs, for instance, with chickens and birds. Again, most research identifies from the young kids, and Carey and Steppens in particular, planted this seed, and it's been followed through and supported by a range of researchers since. They look at eggs. And eggs, for instance, are not identified by younger students. It's really only up to about age 9 or 10 that they actually start to identify them with life. And trees spawning fruit, for instance, a really interesting split here. If we look at what young children say, they don't identify a tree as being alive until it's spawning fruit. So the best thing for us as teachers to know is when student scientific framework shifts. What happens at the early stages? What happens at the later stages? What happens in the middle? And what impact is our language and the language of the novice having on creating detailed scientific understandings? We need to ask ourselves, can the teaching of science help learners to build up their concept of living organisms by asking them to focus on the similarities shared by different classes of living things? Now, historically, there's different approaches to teaching science. One focuses on difference and the other looks at similarity. And well, what we're going to present here today is really an argument for focusing on similarities. if we can build clusters of similarities, as Ludwig von Wittgenstein pointed out back in the 1920s, if we build those clusters of understandings, we create the word images. If we create the word images, we create the concepts. If we create the concepts, we create the schema. And if we create the schema, we are creating the frameworks. And this is what we're trying to do. So when we look at it, what does it mean to be alive? Imagine yourself in your classroom and a student asks you, for instance, or tells you or communicates that fire is alive. It's an interesting response, isn't it? It's got movement, let's face it. That's what the research dictates. You know, if it's got action, it's got movement, it's alive. So the only way you can actually respond to that is to have given it some prior thought. Build up a set of criteria about what constitutes a living organism. And then for each of those criteria, decide whether it's necessary or an optional characteristic. So here's a little example, a pretest, something we do. When you're looking, for instance, at the 5 E's or POE, one of the first things you do is you go in and you ground existing knowledge. You locate the students. Okay. And here, for instance, we've got a range of objects, a pretest. The earth, fire, the sun, a virus, a daffodil. Student number 1 says the earth is living, it moves, it grows. Student number 2 responds because it's changing. Fire is non-living. Student number 2 says it's living because it runs on fuel and it's full of energy. You can see the metaphor. The sun is living like the earth, the student living because it changes all the time. So you can see the concept coming through for student number 2 here. It's got to do with change and evolution, change and movement, change and development. A virus, again, student 1 says it's alive because it eats, it moves, it grows and it dies. It's got a life cycle. Fairly elaborate concept. Student number 2 says it's living because it can attack you. A daffodil, it still lives, but after a while it dies. And response number 2, it's living because it drinks. And again, in response number 2, we don't see any sort of concept here of time, sequence, evolution process or cycle. So when we ask a child, what does it mean to be alive? The REALS project coming out of Deakin University identifies from Titler et al. And a group of researchers out of Deakin, and they point out quite clearly that children will vary in their concept of what constitutes life. A little example: why not do an applied experiment in your own classroom? Okay, it could be in your schoolyard, and this is what we're arguing. Science is local, science is at hand. A freshly picked papaya, a little constructivist experiment. Consider the similarities, note the similarities over time between 2 papaya from the same tree. The students can be there, they can watch you pick them. Leave one on the trunk, pick one, both are green. One is placed on a sunny shelf. See what happens, it changes from green to yellow at a similar pace to the one still attached to the tree. We can then look at different rates. You can see the space here for charts, for charting, for live data gathering, for data mapping. Both would eventually show signs of water loss and decay. We'll start to see some physical change in their appearance. However, even when mouldy spots appear, the whole papaya tissue would not be rotten. So again, they're going to appear at different rates. We may use photography, we may use a whole sketching, we may use diagrams. Okay, we can weigh them to see whether there's any change in the physical mass. And finally, parts of the flesh of the picked and unpicked fruit are able to maintain themselves in a condition not dissimilar to that of a young pawpaw ripening on the trunk. So we're going to be able to look and see what those different processes are. So it's creating an observation framework. Animals. Bell. Some of you will know about Bell's famous experiments. She investigated which organisms children thought of as animals and why they thought. So again, looking at that similarity. And down here in the bottom right-hand corner, we can see the little plant structure and animal cell structure. We know they're different. They look physically different, and they contain at least 3 different properties. If you're not sure what they are, then jump on Google and find out, because you may be asked that question. So Bell investigated which organisms children thought of as animals and came up with the following. Mammals, such as cows, present little difficulty. Whales are a little bit problematic. At different age groups, some students actually don't think of fish as animals. Organisms, spiders and worms, are regarded as animals by between only 20 to 50% of primary school children. Interesting, isn't it? Children aged 5 to 6 are more likely to think of spiders and worms as animal biologists' view than children aged 9 to 10. So as they get older, there's a shift in their paradigm. Why? Because when we get them into schools, we start to fool around with their metaphor. They gain knowledge of biological groups, reptiles, insects, types of worms. They lose the inclusive animal concept. In other words, they begin with a global idea of what an animal is in age 5 to 6. And by the age 9 to 10, that tree has been shaken. Certain species have fallen out. Certain characteristics have fallen out. And as a result, by the time they're at years 9 to 10, their framework is probably less robust than it was back at age 5 and 6. So when we focus on differences, okay, what we're doing is we're dismantling the learner's framework. Okay, so what we've got to do is find a way of embracing similarities. If we're going to help them to lay down the foundations of scientific concepts, we must build on those similarities. So invertebrates in the school ground, a fantastic little unit that you can do. As you can see here, we've Tytler and his teams and cohort designed this from scratch, and it's known as the RILS project in science research. And what they do is they talk about using representation in the learning of science. So, for instance, they begins with a pre-test up here in the top left-hand corner. Debate the characteristics of living things. Work on computers to construct interactive questionnaires about what make up living things. They discuss what it'd be like to work as a team of scientists, finding out what living things exist in their school grounds and why. Student predicts what they might find in their habitat. Now, here's an assumption that every school ground has its own habitat. And again, part of this investigation is to demonstrate, explore, and prove that it does. Not only to prove, but to define and elaborate what that ecosystem might be. So they report their observations back to the class. They discuss inferences to what they think might be going on in those habitats. So we're starting to see some action. Students conduct habitat explorations, okay, to make a poster. What is in the habitat? And you can see the emergence here of a food web, a hierarchy, a set of relationships between the animal, the environment, and its particular living needs and wants. Students further explore in step 4 the habitats to answer research questions. They record environmental conditions, note the number of each invertebrate found, and drew and annotated drawings to describe body structures and behaviours. Very simply done. All you need is a jar, some mesh, an elastic band. You can catch some invertebrates, and once you've got the invertebrates caught, you can then actually begin the process of collecting and categorising these invertebrates. Students collected one invertebrate and observed it with a view to constructing a model, a 3D model, that would represent the movement or feeding of that invertebrate. They used digital microscopes, made annotated drawings and time sequence drawings, and described how the body structures and behaviours of the invertebrate adapt to the habitat. Okay, so they're hypothesising here. They then went on at stage 6 to conduct group-generated activities using mealworms and stick insects to establish ways of describing body structures, movement and feeding. So notice here, two creatures that obviously have the common term of living together. Okay, they share that common framework. But what we're talking about here is looking at different ways of describing body structures, movement and feeding. So we're creating 2 different examples, 2 different contexts, and also for testing what their behaviour may do. And for instance, again, focusing on similarities, a Venn diagram, what do they have in common? And therefore we can also list legitimately what they have different in terms of their characteristics. Students developed and presented posters giving an account of living things, okay, and discussed some preliminary ideas how the animals and plants might interact. They then start having a poster show, and they captured selected invertebrates, observed them and recorded body structures, drawings, digital photographs, all the work that Joseph Banks used to do, all the work, for instance, that Charles Darwin used to do. These kids are what Piaget would call, the child scientist. They use digital microscopes and the internet to identify and classify. They then release the invertebrates where they had been found. They return them to their ecosystem. Students used scrap metals, building blocks, modelling clay and plastic straws to build a 3D model of the invertebrate that they then used to represent as accurately as possible how it moves or feels. And some of these took the form of puppets. They don't have to be elaborate. They can be puppets, hand puppets. And the final point, of course, a post-test. Okay, how are you going to set up an evaluation tool to explore student knowledge about concepts related to animals in the environment, the nature of science, and science investigation? So what will that tool look like? Piaget, 1962, the child as a scientist. And we can look here, for instance, that we can send, the child out into the playground, out into their own school ground. How are animals in this environment suited to political, physical aspects of it? And here are some of the things they can do. Okay? Plan and side sketch. Sketch plants and leaf litter. Monitor the percentage of plants covering the species and how many of them there are, their frequency. Draw to show temperatures at different points in the yard or in the seasons or in the year. Or depths and moisture and descriptions of the environment and its variations. You can move on to the animals that live in this particular environment and where exactly do they live. So we can look at some of the biodiversity here, the populations, animal numbers. We can actually start to do chart and graph. We can come up with scales, frequency. We can look at food and food sources. How do animal structures and behaviours support their survival? Again, moving further in here, we get the 3D model to represent a particular feature of movement, an invertebrate of their choice. And finally, how do they interact with each other? And here largely we know the animal kingdom is based on chains and webs. So the students move from an identification of the animals in this environment, their interrelationships with the rest of the environment, the plants, okay, and other habitat features, ultimately to being able to come down here to have a look at the hierarchy and the organisation of that environment. So their elaboration of scientific concepts is really gaining ground. The child is very much a scientist in the pure sense of the word. Reasoning and learning. We can use generated questions, okay, generated by students from a pre-test, post-test response. So we can actually send students out there to have a look, for instance, at how many ants get eaten by an echidna. Okay, the echidna may be, for instance, in a tank. It may be in its own little pen. It may not be an echidna. It may be a different species of creature. And for instance, they go through, make some observations, and then ultimately at the post-test they come up with a definition, a little map of an echidna. The student writes in its own ideas, okay, and then superimposes some data constructs over the top of it. So we're starting to see, the modification of knowledge here. We're starting to see the student move along that continuum. And of course it's multimodal. It's textual, but it might be written or also spoken. It's visual. It can be diagrams, 3Ds, animations, simulations, photographs. We can get the students to do their work in photographs. If they're really clever, they can actually take a photograph and shoot it back to the laboratory, which could be the classroom, where you have a team of students putting together observations about what's going on in the ecosystem, in the environment, at that very moment. And here you can do a time and motion study. Here's what's happening at 9 o'clock in the morning, here's what's happening at 10:30, and students can simply go out, take photographs, and email them back to home base or send them via mobile phone. Mathematical formula, graphs and tables. We can see all the possibilities coming up here in this particular diagram. And we can also do role plays, gestures. Okay, we can look at how animals interact. We can mimic animal behaviours. Put together a play related on the animal environment. Okay, it's a whole range of things that we can actually do. More importantly, if we use this approach, we're talking about shifting students towards purposeful observation. The whole motion here is action. So why do animals move the way they do? We can challenge learners to conduct scientific observations. And students, they have the agency in an action-based classroom. If everything's active, things are moving. There are representations of science everywhere around them. There's graphs, tables, drawings, reporting. There could be a wonder wall at the back of the room. There could be a discovery table at the front of the room. All of these are reasoning tools, and they would all be built around the purposeful observation of what the students were doing out in their ecosystem. So we can see they're coordinating ideas, they're building evidence in scientific explorations, and they're creating knowledge and generating learning. And as you can see, an important step here is conceptualization. Okay, what do the bugs look like? Do you have a magnifying glass? Okay, break the bug down. How many legs? What are its key features? What are its colors? Draw your little map. Once you've drawn your map, You can then move into the modelling process. And by doing this, look at the materials chosen by this student. Pipe cleaners for legs, a fairly good replication. Have a look at the body. Okay, again, a fairly the scale. Yeah, we can have a look at the mathematics. We can have a look at all of the things going on behind these particular activities. The student making sense of these creatures by exploring and interacting with the creature's environment. A little task that you could use. Okay, and here is the one that's been mapped throughout this discussion. Select an invertebrate of your choice. A really good task to give your students from the habitat around your school. Draw it in as much detail as possible. Use the magnifier. Draw a sequence of drawings to help you build a model, okay, to help you translate that invertebrate into a 3D understanding. So you've got to capture the invertebrate in a container. Here are the instructions. Conduct and write a paragraph to represent how it moves and feels. So we're looking at the literacy component. Use the internet. Again, we're looking at the observations, so the research component. And ultimately we're moving into where we're building the 3D model. Paper, wire, pipe cleaners, elastic, plasticine, containers, all items, all materials that are readily available in a primary school classroom. We may finish up with something like this. Here is a student who went through these stages: drawing, identifying the creatures, modifying and refining aspects of movement, development cycle. You can see the, if you like, the food web here. Here, for instance, we've got to start to convert that information into understanding and knowledge. We've got the hierarchy of the environment, the ecosystem, and it's starting to unpack here. The student's starting to make some observations and some inferences from that process. Finally, the student is converting, okay, modifying this into knowledge and understanding, okay, where we actually have some empirical observations of this particular creature in its environmental setting. The task given to you this week, Activity 1, was the food web, which really is just an elaborate version, a nice textbook version of what this Year 5 student has done here. So, here we have the food web, a set of interrelationships and connections. Here we have a food web, a set of interrelationships and connections. So the science we're talking about is readily accessible to the students as much as it is to the teachers. It's common, it's explicit, and it's shared. The food web activity coming from Loxley, if you have a look, who are the producers? Now, obviously you'll have to go and look at this this activity to understand these terms and to work with them. I'm not going to do that, that's your task. But you do need to, when you're talking to your students, remember right back at the start of this presentation I talked about having a concept map. Okay, it's really important to lay that down. Who are the producers? What is a producer? Who is a first order consumer? And who is a second order consumer and higher? Now, I don't know whether any of you caught the story last week about a blue-tongued lizard down in Sydney who had fallen into a pool, a swimming pool, and the family came home to find the lizard what they thought was to be dead. So they fished it out with a pool net, put it on the side of the pool, and instantly, for some strange reason, began to apply CPR to this blue-tongued lizard. So they were literally had it on its back and they were pumping its little heart. And of course, after about 15 minutes, they realised with their two little fingers crossed that, this wasn't working. And all of a sudden, the blue-tongued lizard's its eye started to move as they were just lifting it up to go and bury it. And they almost buried it alive because the blue-tongued lizard did make a full recovery. And of course, they're now, they've actually still, it's living with them, and they're actually giving it some tonic and food and some nutrients to help it recover. So a really interesting story there about, interactions in an environment. That was in the ABC just last week, so if you do look at the ABC page, you should be able to track it down. But we've got the first order consumers here. The water boatman, right down to the swan helmet, the diving beetle. The second order consumers, who are they? What is the relationship? Now, when you talk to your kids, you need to have this worked out in advance. Okay, you need to work with your students and again, have your inclusion principles. What is a producer? Why do I include them in that category? Your exclusion principles. Okay, the water boatman is a first order consumer. Why do I include him in that category? Why do I exclude him from this one? The blue-tongued lizard, why do I include her as a second-order consumer? Why do I exclude her from a first-order consumer? Now, if you have your framework clear, your learners, your students, are going to be able to rebound off you, like throwing a ball against a wall. And this is how science concepts are developed. The food web, we gave you a couple of questions here this week. Okay, which of the organisms contain chlorophyll? Are they producers or consumers? How do you know? And we can start to see and explore and hypothesize. Okay, so what contain algae, reeds, water ribbons? They're all producers, okay, because they're producing chlorophyll. This photosynthesis, which is one of the topic terms for this week. They are able to convert sunlight into energy, thus producing their own food. So they are producing food. Which organisms feed on algae? And we've got a couple listed there, which feed on fish. Okay, and we've got a couple listed there. So you can see the structure here. What you're doing is elaborating relationships, making connections based on a simple food web. Predict what's going to happen to this food web given certain interactions. Learn for lawn fertilizer. Okay, what happens when lawn fertilizer enters an ecosystem like this? Nitrogen, phosphorus are introduced in large amounts. Smaller amounts of potassium and sulfur in minor elements. We're starting to see chemical changes. Now, while this is the domain of chemical science, as a primary school teacher, you're going to be teaching using all strands. So there's going to be interaction between the strands. So we're not going to have one clear picture. So you're going to have to go in with your concepts clearly mapped. Nitrogen and phosphorus cause dramatic growth, okay, and often spurn, for instance, blue-green algae, possibly causing bloom, okay, algal bloom. They can occur naturally, but when nutrients flow into a river, or, for instance, as we're finding, flow out to the barrier reef, the algae can reduce the oxygen significantly in the water, killing stressing fish and other organisms like coral. Okay, and of course, these toxic products can result in processes like bleaching. Okay, and of course, for man-made processes, drinking and use. Also, if other animals are sharing that water source too. Less water will mean high concentrations of salts and other nutrients in the water. So as we see, for instance, a predicted period of heat or evaporation, we see higher temperatures causes rapid increase in the number of organisms, algae and macroinvertebrates, and the warm water also affect the amount of dissolved oxygen in the water, making the water thicker, slower, and less productive for small creatures to survive in. We asked you to predict what would happen if all of these several things happen. And again, the main reason why we do this is not because we want you to know, because ultimately it's interesting to know, but B, because we really want you to be able to add these sorts of inquiry tasks, these user-generated questions. Okay, throw them into your curriculum mix. The whole aim here is to take a scientific process and to take an ecosystem and to have a look at the biological components of that ecosystem, what they are, why they're defined that way, how they interact, and what their relationships are, and what the nature of those relationships are, and then ultimately what hierarchy emerges, and what happens to that hierarchy if there's a simple or a subtle change. It could be oil spilled. The fish may be wiped out. And for instance, I was back home in Cairns 3 weekends ago and saw several thousand fish floating down Chinaman Creek. Okay, apparently the water levels got too low. The oxygen in the creek, according to the Primary Industries, Department of Primary Industries, the oxygen in the creek, actually creek water, got to the level where it was toxic for the fish, and the fish actually died. As a result, of course, we had an outbreak of dengue not long after that down in Cairns. So we're starting to see the implications of this ecosystem. House owners or farmers, if we look at the barrier reef, followed by big rain, engage in fertilizers, or fertilize and then the big rain comes, the cyclone. The weather is warmer, less rain falls, we have a dry period. Oil forms. Water birds, which rely on fish as a food source, move to another area. So what happens? All of these are in really, really good contextual questions. And by throwing these questions to a student, you are throwing them the tools to become a scientist in their own environment and ecosystem. Hierarchical concepts, the decomposers. Now again, if you're going to be talking to your students about biological relationships, you will know what a decomposer is. What organisms decompose? Where do they live? What do they do? Indicate where they fit into your food web. So you're getting this constant elaboration, this evolution of relationships and dependencies. What tasks would you divide, a hypothetical question, what tasks would you design for your learners in your classroom to help them explore the concept of decomposers? Think about that. The fungi and the bacteria, embedded in the soil, decaying plant and animal matter. They're able to process the larger organism, breaking them down into simple components which they use as food. The simple components are expelled as waste and returned to the food chain, where they serve as nutrients to support forest growth, support plant growth, and also feed certain species. So again, we're looking at the cycle, the continuous cycle. Building concepts in a food web. Again, you can take it a level further. Find a food chain, identify 4 organisms, place them in a pyramid with the producer at the bottom and the highest order consumer at the peak. Okay, and then observe. Estimate the numbers of organisms for each level. First of all, make a guesstimate. Then have a period of observation so that you can confirm your hypothesis. Okay? And we can see, for instance, that we may have too many predators. We can then infer what's going to happen. We may have, for instance, consumers at the second level. We may have consumers at the first level. Okay, we may have our producers at the very bottom, and we can have a look at the impact of changes even amongst those 4 links in that food chain. So again, we are throwing the observation, the prediction, the inquiry back to our students. Now we can take it further. We can look at our school environment. And for instance, we can have a look at who the tertiary, the secondary, primary, and the producers are within a little school ecosystem. It's not hard to do. We can base it on our observations, a week of observation. What creatures do we see? What evidence of creatures do we see? For instance, we may not see a white-tipped rat or a marsupial rat, but we may be able to discern some evidence around the school. We may talk to cleaners, janitors, gardeners. We may talk to a whole range of people, bring in a pest controller or animal observers or whoever it is we decide is part of this network that can tell us a little bit about the types of creatures in our ecosystem. And you'll find generally a snake handler will be happy to come in and bring some very amazing pythons. We can then set about getting the students working. The cricket, the birds, the owl, the plants. We can have a look at our own hierarchical systems. And these are all key tasks. It doesn't have to be a hypothetical system. When we looked at the pond and our last exercise, it doesn't have to be something hypothetical. It can be something that actually exists and exists around your students. So you can take them out and immerse them in it. When we look at this, we're talking about integrating conceptual understandings, inquiry skills, and scientific values and attitudes by using investigations of our own school grounds. So for instance, we can do an activity, random hunting, and we can go out and look at many plants and animals coexist, okay, but we don't think they interact in any significant way. So we're challenging that. So how do we do that? We apply certain inquiry skills. We observe, we sort and categorize, we report on activities using. Our second activity, directed hunting. Doing this by direct hunting, by collecting a particular animal. We're focusing in on an animal, and we're actually then using that focus to explain, how the invertebrate supports a diversity of other animals with different characteristics. So we can start to tune into the patterns and animal type and behaviour. The groups data. We can actually look at animal groups and diversity in simple block and column formats. We can record observations using simple drawings. And this helps the values. We're building interest. We're building fascination. We're creating awe. A variety of invertebrates. Concern for the welfare of larger animals. Direct hunting. Plants. By doing that, we can explain that plants and animals interact with the habitat. Animals use different areas of habitat. Plants have a variety of features. We can develop drawings appropriate use and key. We can pose questions and suggest observations. And the plant is seen as an important entity in its own right. So the value is we've begun focusing on animals as an entry point. Okay, we can then look at invertebrates as a subcategory of animals. And now we can look at plants as a subcategory of the habitat. And so on and so on. We can look at selections of animals for closer observation, detailed representation, modelling, and design of an organism to live in a particular habitat. And here's where we can get a little bit science fiction if we want. We can design a creature that may live, for instance, on an alien planet. It may appeal to some kids, it may not. Understand that plants and animals exhibit a variety of structures and behaviours. Okay, use conceptual knowledge to make relevant observations. And finally, habitats are seen as interesting discovery places. Look for details and features of organisms, and it gets us to ask questions about the purpose of the animal and the ecosystem. The last point for this week really is the setting up of mini terrestrial habitats to study details of behaviour, animals and interactions. And you may, for instance, have in your classroom, you may establish a tank. And that tank, for instance, may feature an animal or an insect population. It could be even an animation. It could be a simulation of your ecosystem based on the information you've collected, the sketches and models that you've built. The conceptual understandings, we can look at how organisms have structural or behavioural features. Plants and animals interact. Food and energy relationships can be mapped. The science inquiry skills, some relevant variables. Use scientific knowledge to explain investigations. Find process information. Draw conclusions by relying on data, evidence-based judgments. And report on the results of investigations in a variety of formats. And all, of course, is underpinned by values and attitudes. Curiosity. Testing ideas. The welfare of the ecosystem. And of course the extension of knowledge and inquiry. Now, at this point, I'm going to just direct you briefly back to TPAC. Last week we talked about your technological knowledge, and as a result we've given you a second assessment task. This second assessment task is actually requiring you to use technology, and out of that technology to apply your content knowledge and your pedagogical knowledge to come up with 2 digital learning cards. Now the assessment task 2, I've been on the forum this week, and quite a lot of the early childhood teachers and early learning teachers are talking about the fact that in the early learning framework there is no science curricula statements and no elaborations, and there are not. I mean, that's a clear fact. It's a generic focus, and that focus is very much on the psycho-technical and social development of the child. So what we need to do is hone into that. So you need to do 2 cards. One clearly can be in 0 to 3 classroom, in the P3 classroom. That should be easy because that is governed by the ACS. Those curriculum frameworks are there for you. Working backwards from those curriculum frameworks, we just need to clarify this question. This question is not about the age of the student or the system in which the learner is located, early childhood learning years or the early childhood classroom. This question is actually about the pedagogy. So pick 2 pedagogical strategies. Is it the 5 E's? Is it POE? Is it inquiry-based learning? Is it problem-based learning? What are you going to call it? Pick 2 of them and demonstrate using a digital task card how that works in the early childhood years and also in the P3 years. The ACS is useful, but it's not essential to help you complete that task. It's useful. But working back, you're going to have in the early childhood years very much a focus on the child as being social, as play-based learning, as literacy, basic numeracy, and also on problem solving. So you're going to have those strong features in the early years, as distinct to the ACS, which becomes slightly more academic. So you don't need the ACS in the early years to complete this task. You have a range of pedagogies on hand for you to draw on. And again, I point to some of the prompts we've given you. Gregson lists some ideas. Again, on 257 to 262, he lists some resources. Loxley has some fantastic ideas you can adapt. Some online simulations and games. Okay, now you don't have to make the game. You can simply link to it, and the pedagogy is the context in which that game is introduced and which students use it. So it's really important. You do not need to go to the trouble of building this resource. And please, get lively on the Q&A forum. This is the time of year when you need to work smarter, not harder. Once upon a time, 30, 40 years ago, education was about competition. People competed for A's and B's and C's or high distinctions and credits. Nowadays, we are only as smart as the people we work with. We're all part of that infamous palindrome. Think about birds. A pack of 5 birds can fly higher and faster and quicker than a single bird can. So this is not about competition. This is actually about collaboration. And then, once you've collaborated, take your learnings and translate them. So get onto the forum. Don't work away in the privacy of your own lunchbox. Please, connect with others. Start posing some questions, start sharing some resources, because if you're wondering how to solve a problem, you can be guaranteed there are at least 20 other people with the same question. Work smarter, not harder. I will catch you all at tomorrow's Zoom session. Thank you.